Hello, everyone, and welcome to today's webinar, Lean Lab Testing Quality Attributes of Water for Pharmaceutical Production. I am Sydney Janetta with Sievers Instruments by Suez, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Suez Water Technologies and Solutions. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like now to introduce our speakers. We have Kaitlyn Vap, the lead product application specialist for TOC and conductivity. Hayden Skalski, the lead product application specialist for bacterial endotoxins, and Briana Nunez, lead researcher and product specialist for microbiology. Kaitlyn, Hayden, and Briana, you may now begin your presentation.
Hello, everyone. My name is Kaitlyn Vap, and I'm the lead product application specialist for life science at Sievers. I'm joined by Briana Nunez, the lead researcher for microbiology, and Hayden Skalski, the lead product application specialist for Endotoxin. Today, we're going to be talking about testing quality attributes of water for pharmaceutical production and how technologies capable of doing so have evolved to lean out laboratory testing practices. During this presentation, we'll cover content around quality attributes. We'll first provide an overview of what quality attributes exist, including conductivity, total organic carbon, endotoxin, and bioburden. We'll then talk about why and how we test for those attributes, eventually getting into how the testing of those attributes have evolved to optimize efficiencies. We'll then wrap up with a conclusion and leave time at the end for any questions. So let's start with defining pharmaceutical water quality. The country-specific pharmacopoeia organizations regulate quality standards of the pharmaceutical industry. The United States Pharmacopoeia, among others, require the testing of conductivity, total organic carbon, or TOC, endotoxin, and bioburden as it applies to water for pharmaceutical production. Evaluating the purity of water as a raw ingredient of pharmaceuticals is one way pharmaceutical manufacturers build quality into their processes, and it's going to be the focus of this presentation. The primary reason pharmacopoeia organizations mandate quality attribute testing is for risk mitigation purposes. While pharmacopoeia is mainly concerned with patient risk to pharmaceutical manufacturers can be broken down into three categories: patient, process, and business. Patient risk being associated with the efficacy and safety of administered therapeutics, process risk being associated with contamination events, and business risk being associated with costs incurred from potential recalls or out-of-specification results. By monitoring quality at various stages of the process, including raw materials, it's easier to mitigate these three categorical risks. Traditional methods for testing have been laboratory-based. The traditional methods were also coupled with labor-intensive techniques and slower testing or assay response times. Alternatives to laboratory-based testing are at-line and online testing, which begin to lean out steps of the testing process. At-line testing can occur in a direct environment in which the sample is pulled for testing, eliminating the need to transfer samples from a production setting to a laboratory. Online testing is one that occurs in real time as water passes through an online instrument, eliminating the need to isolate and transition samples to the laboratory. As these apply to lean laboratory practices, lab, at-line, and online instruments can all be considered lean lab instruments. Lean lab is a relative term, with traditional testing methods serving as a baseline. With that in mind, technologies that emerge to improve testing efficiency and productivity, even if that's a new lab instrument, can be considered a lean lab testing method. After we talk about some more of the traditional testing methods, we'll discuss what lean lab instruments currently exist for conductivity, TOC, endotoxin, and bioburden. So let's start with conductivity and TOC. What is conductivity? Conductivity is a measure of the ability to pass an electrical current through a substance. Inorganic chemicals and salts present in a substance can alter conductivity readings. As inorganic ions can come in the form of cations, which are ions that carry a positive charge, and anions, which are ions that carry a negative charge, they have the ability to act as a carrier for electricity, which can then be correlated to a conductivity reading. The amount of dissociated ions present is a function of pH and temperature. USP monograph 645 requires water of pharmaceutical grade to fall under 1.3 microsiemens per centimeter at 25 degrees Celsius. Carbon dioxide readily dissolves in water and can dissociate into ions. These are known as intrinsic ions, while chloride and ammonia can be indicative of extraneous ions, both of which can contribute to conductivity readings. Why do we test for conductivity? USP 645 models their 1.3 microsiemens per centimeter at 25 degrees Celsius limit from the acceptable amount of carbon dioxide, chloride, and ammonia ions present, as these ions can have a negative effect on the water's chemical purity and suitability for pharmaceutical production applications. Carbon dioxide can relate to the amount of total dissolved gas present, while chloride and ammonia can be indicative of chemical contamination at high enough levels. Chloride can damage equipment at high concentrations, and both chloride and ammonia at high enough levels can be harmful to human health. USP standards fall significantly below the levels that would be harmful or toxic to human health, but ultimately the mandated monitoring of conductivity stems from inorganic ions and salts posing risk to patient safety. Let's move on to total organic carbon, or TOC, starting with what is TOC? From a mathematical perspective, TOC is total carbon minus inorganic carbon. TOC is an indirect measurement of the amount of carbon dioxide formed through an oxidation reaction. This is differentiated from inorganic carbon, as inorganic carbon is all intrinsic carbon dioxide to the water, while TOC comes in the form of organic carbon molecules that have been oxidized. Carbon dioxide content related to TOC can be traced back to the amount of original organic compounds present. USP monograph 643 is responsible for regulating TOC content in pharmaceutical water, and it's required to fall under 500 parts per billion. Why is it important to test for TOC? The amount of organics present can be derived from chemical contamination or bacterial contamination. Sources can include biofilm and distribution system contamination. TOC also has a qualitative relationship with microbiological activity that can lead to increased endotoxin and bioburden as organic compounds act as nutrient sources for bacteria. Between TOC byproducts that can impose danger to human health and heightened bacterial contamination that can pose the risk of disease, TOC became a mandated quality attribute. Now that we better understand what conductivity and TOC are and why we test for these attributes, Hayden, could you explain a little bit more about endotoxin?
Thanks, Kaitlyn, for that wonderful introduction and for kicking us off. Today, I'm going to talk about how we currently test for endotoxin, what is currently out there for the techniques for detection of endotoxin, why we test for it, and also how we can lean out the QC lab in terms of endotoxin testing. So what is endotoxin? Endotoxin is a type of pyrogen and is a component of the exterior cell wall of gram-negative bacteria, such as E. coli. Endotoxin is a lipopolysaccharide, also known as LPS. The LPS structure consists of a lipid A portion containing fatty acids, a disaccharide phosphate, and core polysaccharides, as well as the O antigen. The lipid A portion of the LPS is the main cause of the molecule's endotoxin activity. Now, human immune cells see that lipid A portion of the LPS as bacteria or a foreign invader. And that foreign invader, or that little trigger that the human cells see from that lipid A portion, will stimulate a response to fight off these intruders. Endotoxin is present in food and water and pretty much everywhere else. So it's extremely dangerous if it's entered via the bloodstream or intrathecally, which is the spinal canal route. Endotoxin can be introduced a few different ways, which include injectable drugs such as vaccines, medical devices, surgery, or bacterial infection. So why do we test for endotoxin? Again, endotoxin is ubiquitous in nature, meaning it's pretty much everywhere, as we learned in the last slide, including food and water. Now, if certain amounts of endotoxin are introduced into the bloodstream or spinal canal via a parenteral drug or medical device, we can develop a fever, shock, organ failure, or worst case scenario death. Due to the possibility of serious illness or death, all injectable pharmaceuticals and implantable medical devices that come into contact with the bloodstream or spinal canal must be tested for endotoxins to ensure patient safety. Even after sanitizing and cleaning objects or equipment, the bacteria may be killed off, but endotoxin will remain behind and be present. This is why it's super imperative to test for endotoxin throughout the manufacturing process. Mostly all parenteral drug and manufacturing and medical device companies will use water for their products. Some of these water uses will include drug production, so fermentation, lysis, purification, also known as downstream cell culture, upstream activities, cleaning validation, cleaning of vessels, containers, bioreactors, glassware, buffer and drug formulation, pre-fill and finish, lyophilized drug reconstitution prior to the drug administration, medical device manufacturing, and medical device cleaning. All manufacturers of parenteral drug products, which include intravenous, intramuscular, intrathecal, and also medical device companies that make med devices that come into contact with blood, will test for endotoxin. Also, veterinary and animal health products that come into contact with animal blood will also be testing their products for endotoxin testing. Now that we learned a little bit more about endotoxin and why we test for it, Briana, can you tell us a little bit more about bioburden?
Of course. Thank you so much, Hayden. It was a really good description of endotoxin. I'm going to get into what is bioburden and why we test for bioburden. So what is bioburden? The term bioburden is commonly used to describe the total population of viable microorganisms present on a surface or in a product. Viable just meaning living. So a microorganism or a microbe is a living organism that can be only observed through a microscope. Common microbial contaminants or bioburden found in pharmaceuticals or biopharmaceutical production facilities are often bacteria or fungi. So bacteria are a single cell prokaryote that are very adaptable to all environments they come into contact with. They come in different shapes, they come in different sizes. Some are gram positive, some are gram negative, and some are capable of forming spores. Fungi is generally broken down into two groups as a bioburden that's seen in production facilities, and that's molds and yeast. So a mold has a very fuzzy appearance. It can be orange, black, green, purple, and found in several different types of shapes. A yeast is a type of fungi that contains a single cell and can cause infections in individuals who are immunocompromised. So each step in the process of production inside a pharma facility will have bioburden limits. So a raw material or a starting material will have different bioburden limits compared to a final product. So the ability of microbes to survive and actually thrive in some of these conditions is generally a thing of beauty for microbiologists. However, the ability of microbes to be able to do this is incredibly detrimental to production facilities. So the question is, why do we care about bioburden and why do we test for bioburden? So in January of 2012, the American Pharmaceutical Review published an article detailing recalls involving microbial contamination. Of the 103 cases presented, 34 of which were caused by Burkholderia cepacia. So Burkholderia cepacia, or Burkholderia cepacia complex, is a gram-negative bacteria that tends to mutate and adapt very quickly to its environment, even in environments that are pretty nutrient deprived. Cepacia can metabolize organic material present in in aquatic environments, such as ultrapure water loops. Cepacia can cause very serious respiratory infections in individuals who are immunocompromised. It's known to be an opportunistic pathogen that can cause morbidity and mortality. So one of the reasons we test for bioburden is to ensure the health of consumers. Bioburden testing is also used in the manufacturing process as a monitor. So sampling and testing will occur throughout the process to assess the quantity of bioburden present starting in raw materials, moving towards in-process materials and intermediate products, and also the final product. So as an example, if a raw material or a starting material was used and it was contaminated, the contamination would follow all the way through. So your final product would also have the same bioburden contaminants that were originally in that starting material. So factors that can influence bioburden levels are generally broken down into three different categories. There's equipment, environment, and also product. So is the environment that you're working in disinfected properly and regularly? Is the HVAC system maintained, or is it blowing microbes all over your workstation? Is the equipment used in the production maintenance regularly? And then once a product is produced, is that product being stored correctly? So all of these different things can influence bioburden levels in a production facility. I'm going to hand this back off to Kaitlyn, and she is going to talk about how we test for TOC and conductivity.
Thanks, Bree and Hayden, for those explanations on bioburden and endotoxin. As Bree mentioned, we're now going to shift gears and get into conductivity and TOC, specifically how we test for these quality attributes. So starting with conductivity. Conductivity is the inverse of resistance, and resistance is the opposition of electrical current. It can be correlated to the amount of voltage applied over the current, which is the movement of positive or negative electric particles. With resistivity, conductivity can be calculated. To measure for resistivity and correlate to conductivity, instruments typically operate off of two electrodes with a voltage applied between them. The drop in voltage caused by resistance can then be used to calculate conductivity within an algorithm intrinsic to the instrument. Moving on to TOC, how do we test for total organic carbon? All instruments that quantify TOC have a few fundamental properties, including they oxidize organic compounds to carbon dioxide. The mechanism in which they do this might differ, though. It can include UV oxidation, combustion, or supercritical water oxidation. Once the organic compounds have been oxidized to correlate to the total organic carbon content, the second fundamental quality is that instruments measure that resultant carbon dioxide. Again, the mechanism in which they do this might differ, though. So there's direct conductometric, membrane conductometric, or non-dispersive infrared sensor technology, or NDIR technology. With this, inorganic carbon can either then be measured or it can be a value that's factored into the final TOC calculation through an instrument algorithm that has the ability to predict the amount of inorganic carbon present. What's important to note to maintain compliance is that carbon must be differentiated and detected according to USP 643. This means, number one, you must differentiate between inorganic carbon and organic carbon. And number two, carbon and only carbon must be the species detected. So the analyzer must be clear of any sort of ionic interference. This is ultimately what differentiates membrane conductometric technology from direct conductometric technology. In the diagram to the left, the animation is a depiction of direct conductometric technology, and the right is membrane conductometric. Membrane conductometric TOC analyzers have the ability to differentiate between inorganic carbon and total organic carbon, while also measuring only carbon with a selectively permeable membrane to filter out any interfering ions that could cause over and underreporting that's commonly experienced with direct conductometric instruments. So while direct conductometric TOC technologies may be beneficial for process monitoring, it's recommended to pursue membrane conductometric technologies for process control and release applications, since they're in compliance with USP 643. So now that we've gone through how to test for conductivity and TOC, Hayden, can you share a little bit more about how we test for endotoxins?
Thank you, Kaitlyn. So I will go ahead and discuss how we test for endotoxins currently in the industry. So how do we test for endotoxin? Bacterial endotoxin testing, also known as BET, is an assay for measuring active endotoxin in water and products by combining a sample with limulus amoebocyte lysate, also known as LAL reagent, and measuring the resulting proportional reaction. This LAL reagent contains an aqueous extract of circulating blood cells, also known as amoebocytes, from the Atlantic horseshoe crab here in North America. So there are a few ways these LAL tests are ran, which include gel clot technique, two photometric techniques called kinetic turbidimetric and kinetic chromogenic, which we will discuss in a few slides. The LAL reagent is derived from the horseshoe crab's blood. So the LAL reagent comes from the Atlantic horseshoe crab, which is Limulus polyphemus. The amoebocytes are the blood cells, and the lysate is the cellular extract that is used for endotoxin testing. The enzymes extracted from the blood trigger a reaction in the presence of endotoxins. The blood clotting system contains a cascade of 3 inactive enzymes and a clottable protein called coagulogin. The reaction cascade ultimately ends in a gel, turbidity, or a color change, depending on the method chosen. So the common means for endotoxins testing today, again, there are 3 methods, which are USP 85 compendial. The first we see here is gel clot. So this is going to be a qualitative and subjective method. This gives you a pass-fail test, or endotoxin is present, endotoxin is not present. You're looking for a formation of a gel in a test tube. So your sample and LAL reagent are added to a test tube, which is then incubated in a heat block for an hour at 37 degrees Celsius. And at the end of the hour, the analyst will invert that tube upside down. And if it clots, it means it's a positive, or endotoxin is present. And if it does not clot, it means it is a negative. Kinetic chromogenic, which is also known as KCA, that is a photometric technique. It's quantitative, so it gives you an actual result. And this method measures endotoxin over time for the appearance of a yellow color. Now, we have a colorless chromogenic substrate that is added to the LAL reagent, which causes the yellow color to develop after the cleavage of a chromophore called PNA, or para-nitroaniline. So the more color that is released in the solution with your sample, that means the more endotoxin is present. And lastly, we have kinetic turbidimetric, or KTA. This is another quantitative kinetic method that measures an increase in turbidity or the time taken to reach a particular level of turbidity, also known as onset time. The onset time of turbidity is inversely related to the amount of endotoxin in a sample. Now that we learned a little bit about how we currently test for endotoxins in the industry, I'll pass it over to Briana and she can explain how we currently test for bioburden.
Thank you very much, Hayden. I'm going to get into how we test some bioburden. Bioburden testing as a whole is kind of old. It basically involves growing microbes on an agar plate, waiting for the agar plate to show colonies, and then counting those visible colonies. So the US, European, and Japanese pharmacopoeia have all been harmonized to define bioburden testing. To actually get a result for bioburden testing, you take the total microbial count, which refers to your bacteria, you add it to your total yeast and molds count to receive your total viable count. So the different populations of organisms are assayed differently, mainly because yeast and molds require different nutrients than bacteria. They'll often require different incubation times, they'll require different incubation temperatures, so separate assays occur to assess the entire population. The three harmonized pharmacopoeias have all listed 3 different methods to determine the amount of bioburden present in a sample. So the first test method is membrane filtration. Membrane filtration is the most preferred because it's the most accurate. And what happens with that is when there is a sample passed through a 0.45 micron filter or less, the bioburden is collected on the top of that filter, and then the filter is then transferred to an agar plate. The agar plate is put into an incubator, and at the end of incubation period, colonies are counted and reported. So there's 2 different types of direct plating methods, and of the direct plating methods, there's pour plate and spread plate. Pour plate is the most preferred of the 2 because it has greater accuracy. So a pour plate method uses 1 ml of test fluid and about 15 to 20 milliliters of a liquid agar media. Both the test fluid and the agar media are mixed together. The media is allowed to solidify and then it goes into the incubator. Spread plate method uses between 50 and 500 microliters of a test fluid. The test fluid is added to the top of an agar plate and then it is spread around on top of the agar plate. Most probable number is another test method, but it is the least preferred because it is the least accurate to test for bioburden. It's a statistical method which uses an estimation of viable organisms present in a sample. So the test method involves taking the original solution and dividing it in to different magnitudes, orders of magnitudes into different culture broths. The culture broth is then assessed for the presence or absence of bioburden. Generally, bioburden is reported as CFU per milliliter, which means colony forming unit per milliliter, or colony forming unit per gram, depending on what type of product was used. So I'm going to pass this right on back over to Kaitlyn, and we can start talking about how we can lean out our laboratory.
Thanks for sharing those interesting methods for testing endotoxin and bioburden, Hayden and Bree. Now that we understand traditional methods for quality attribute testing, let's transition to how quality attribute testing has evolved to incorporate these lean lab techniques. And again, we'll start with conductivity and TOC. So real-time testing is an application that's made headway in recent years, but was originally established in 2010, the ASTM guidance document E2656. To understand what real-time testing is, we need to delineate between 3 ways in which online instruments can be used. 1, process monitoring, 2, process control, and 3, process release. RTT, or real-time testing, is the practice of using online instruments for all 3 applications. This requires additional validation, but that inherently improves confidence in all 3 ways to leverage online instruments. beyond just real-time testing's ability to optimize efficiencies. This is effectively a roadmap to integrating RTT into everyday practices for pharmaceutical manufacturers, with 5 phases broken down into subset elements associated with each phase. RTT requires 1, that end users scope the project and organize timelines around project phases. 2, perform a risk assessment with the intent of choosing a technology and placing the technology on the water system. 3, implementing with a series of method and process validation steps. 4, data reporting and handling tools. And 5, keeping up with annual maintenance and redundancy of the instrument. Ultimately, through implementing RTT, you will have the ability to evaluate and ensure quality in real time through trending capabilities seen in the graphic below. RTT implementation also provides an opportunity to expand your process analytical technology portfolio to drive progress for the FDA's PAT initiative. And with RTT, you will have built process monitoring, control, and understanding, all while being able to release water into production in real time. With that, I'll transition it to Hayden to learn a little bit more about lean lab practices for endotoxin.
Thank you, Kaitlyn. So how can we lean out the lab with regards to endotoxin testing? We can do that by introducing microfluidic technology that helps automate the BET assay while remaining fully compliant with the compendia. Microfluidic technology that uses a microplate with embedded reference standard endotoxin, or RSE, with a standard curve range of 50 EU per mil down to 0.005, and positive product controls with every sample will drastically decrease the potential for human error by minimizing that hands-on time. There are several benefits of microfluidic testing, and I will talk about a few of those here with the microplate that we see. So a few benefits will include quick and short setup time, so about a 5 to 10 minute setup time to load the microplate with 21 samples. We have about 89 to 90% fewer pipetting steps in your traditional 96-well plate assay. So it goes from around 242 pipetting steps down to less than 30. We have a 90% reduction in LEL reagent. So we have sustainability in the back of our minds. We take 1 milliliter of LEL reagent for this microplate, and that can test up to 21 samples, where a traditional 96-well plate, you will need around 10 milliliters of LEL reagent. We automated the creation and loading of the standard curve, so we have that standard curve already embedded on the plate. And we automated the loading of positive product controls, and all of your PPCs are already on the plate as well. On top of having microfluidic technology for BET, there are a few more ways to lean out the lab with Sievers Eclipse endotoxin platform. There is a client-server installation, so endotoxin data can be reviewed and signed off remotely with the client-server feature, minimizing the need to physically be in the lab, so you can have less footprint in the lab in terms of employees. And then we have the other reduced footprint in the lab. So the endotoxin analyzer itself is lighter and smaller than traditional endotoxin analyzers for kinetic chromogenic units. So you will have more bench space to do other activities. And then we're going to have a reduced cold room storage and increased sustainability. Now, Eclipse microplates can be stored at room temperature. We have stability currently up to 25 months. So there's no need to store plates in the 2 to 8 fridge and taking up all that space. And also, again, the Eclipse uses 1 mil of LEL reagent per plate. So we're going to have a reduced number of reagents needed to be stored in that 2 to 8 degrees Celsius for testing. Now that we learned a little bit more about the lean lab in terms of endotoxin testing, I will pass it over to Briana.
Thank you, Hayden and Kaitlyn, for getting into some ways we can lean out the lab and different processes to make it a little easier. I'm going to talk about how we can lean out the laboratory for bioburden testing. So new technology is slowly starting to evolve away from traditional plate methods. So traditional plate methods for bioburden detection is accurate, but depending on the organism, it takes days or weeks to get a result. The test method can also be pretty labor intensive. So it requires people going out to sample, people in the laboratory to actually sample and count plates. So there's a new rise and a new interest in RMMs, which are rapid microbial detection methods. So rapid microbial detection methods are technologies that allow the user to get a microbiology result faster compared to traditional culture plate methods. There are 3 different categories of RMMs that include qualitative, quantitative, and identification methods. So if the user receives a presence or absence, that would be considered a qualitative rapid method. If a user receives a specific genus or a specific species of the organism found in that sample, that would be considered an identification method. And if a user receives a result at a numerical result at the end of their test for the amount of bioburden found in that sample, that would be quantitative method. So inside of these 3 different scientific principles are different methods that the industry uses to determine bioburden. So one of these includes growth-based methods. A growth-based method decreases the time in which we can detect a growing microorganism. So some of these test methods include detecting carbon dioxide or changes in pressure. During microbial respiration, microorganisms consume oxygen and produce CO2. So if a microorganism is respiring inside of a closed container, there's often going to be changes in pressure that will result. Viability-based technologies are now pretty common in the industry, and that's because they can differentiate between a living viable cell and a dead cell. So flow cytometry and solid phase cytometry are both commonly used in conjunction with those viability stains or labels. Individual particles are passed through a laser beam, and they're able to be counted with flow cytometry, while solid phase cytometry captures microbes on a solid surface, and then they are labeled with viability stains. Cellular component-based test methods rely on cellular markers or probes that are specific to that microbial target site. So examples would include ATP bioluminescence and also the use of mass spec for microbial identification. Viability stains work very quickly to label and target organisms, making results available within hours or even minutes. So Siever's solution to rapid micro includes viability stains in combination with ultrasensitive flow cytometers. Ultrasensitive flow cytometers are accurate down to a single cell or particle and can return results faster due to the binding efficiency of dyes and stains used. The number one advantage to using an RMM to lean out your laboratory process is you get faster results. So current plating methods are accurate, but they take a long time for results. RMMs can reduce product release times. So instead of waiting for test results, a product can be released to market much faster. By monitoring the production process and receiving faster results, a production site can implement corrective actions sooner and improve the quality and safety of their products.
Thanks, Bree and Hayden. It sounds like there are incredibly innovative lean laboratory technologies that exist for both endotoxin and bioburden. As pharmaceutical manufacturers strive to decrease the time to market for life-saving therapeutics, traditional quality control practices can become a limiting factor. Therefore, by adopting lean laboratory techniques and instrumentation, pharmaceutical manufacturers can improve their efficiency and productivity with real or near-time results and decrease the quality staff's workload, all while maintaining compliance. With faster time to results, the ability to control the process becomes much easier with direct access to data that's reflective of present time points. This improves the capability of ensuring consistent water quality for release into pharmaceutical production. So that brings us to the end of our presentation. If there are any questions on the content covered, we're happy to field those at this time. Thank you. Thank you.
Thank you, Kaitlyn, Hayden, and Briana for your informative presentation. We will now go ahead and start the Q&A portion of the webinar. As a reminder, if you have a question you'd like to ask one of the speakers from today, please do so now. Just click on the Ask a Question box located on the far left side of your screen. We will answer as many questions as we have time for. So let's go ahead and get started. Our first question is directed towards Hayden. When it comes to testing for endotoxin, what about the dialysis water for hemodialysis patients?
So the answer is yes, you should be testing dialysis water for hemodialysis, since the dialysis water will be helping to filter and clean your blood. So we want to make sure that the water we are using for that is super clean. It can cause life-threatening issues if the treatment water is contaminated. So clean dialysis water is crucial, and testing for endotoxins for that is essential to patient safety.
Thank you. Briana, this next question is for you. What applications benefit the most from a rapid bioburden monitoring method?
Great question. So end users have stated that the most valuable applications is at-line monitoring of drug substances and also drug products. That's followed by water monitoring and also environmental monitoring. And another application of interest is definitely cleaning validation.
Great, thank you. Kaitlyn, we'll head to you next. What is the difference between online monitoring of TOC and RTT?
That's a really good question. The main difference is an extra validation component. So as mentioned in the presentation, that implementation phase has a series of method and process validation steps. The method validation steps are standard between online monitoring instruments and RTT capable instruments. That is your standard instrument qualification, operational qualification, and performance qualification packages. When trying to transition from the ability to monitor and control with an online instrument to real-time test and release with an online instrument, there are additional process validation steps that are required for additional confidence, essentially, in that instrument. And what those include is a method transfer phase between the lab-based instrument and the online-based instrument, and then a method comparability study where you're evaluating the efficacy of the online instrument against the laboratory-based instrument, and then also a point of use comparability study where you're evaluating the quality of water at some of your most critical points of use against the quality of water at that instrument that's permanently installed and being used for release. So the final answer there is that those additional validation steps are really what differentiate your ability to monitor versus release water with an online instrument.
Thank you for that description. That's a lot more helpful. Briana, we'll go back to you. What is the recommended implementation of online bioburden systems on water loops?
So an online bioburden analyzer will be typically deployed at the return leg of the water loop. The return leg of the water loop is known as your CCP, or your critical control point. And your CCP is generally indicative of the overall health of the system that you have.
Thank you. Hayden, I believe this is in reference to the Sievers Eclipse. Can I use my current lysate with your system?
Great question. The answer to that is yes. So the Eclipse platform uses commercially available kinetic chromogenic LEL reagent. So which means that if you are running that technique today, you will be able to continue using reagents from your current supplier. So it is available to use from any LEL reagent provider that's FDA licensed today.
Great, thank you. We'll go ahead and keep going with you, Hayden. This one ties into the same thing. How do you validate water samples with the Sievers Eclipse?
So this validation process, especially for water, is super easy. Since water is mainly tested at a 1-to-1 dilution or neat, undiluted, you won't have to make any dilutions. So you can basically just take all of your water samples and test them on the Eclipse. And you can do a simple comparability study to ensure that your water samples are going to be compatible with the Sievers Eclipse. So it is a pretty easy, straightforward process for water.
Thank you. And just a reminder to continue submitting your questions. We still have a few more here, but just to get those in if you have any coming to you. I'll go ahead and go to Kaitlyn. What should I consider to complete a failure mode and effects analysis?
Great question, because this is a really important factor when implementing real-time testing. So when you're considering a failure mode and effects analysis, or an FMEA, in the context of real-time testing, what that is used to inform is where you end up placing the instrument of record, the online instrument, and also which critical points of use exist that should be tested against the water quality at the location of the instrument of record. So with that being said, two main factors should likely be considered in the process of completing an FMEA. And that's one, where along a water loop is representative of the quality of water at every section of the loop. And then two, which points of use are most critical to either the process or the final drug product. So when you're considering where to place the instrument, normally the resultant of an FMEA directs you to place it at the return to tank on a water loop. Because in theory, the water is passed through every section of the loop in which it may have come in contact with something that would have contaminated it. So it should be indicative of the quality at every section along the water loop. And then ultimately, it's up to customers to determine which points of use are most critical to the process and drug product, whether that means a point of use that's commonly used for manufacturing process, a point of use that's being used to dilute final drug product, whatever that might mean for you. There's a guidance document, IEC 60812, that can help you or guide you through the process of completing an FMEA.
Thank you. That was a very helpful description. We'll go ahead and stick with you, Kaitlyn. Can I do RTT with my existing online TOC sensors?
Good question. So as we had discussed a little bit in the presentation, there's different mechanisms in which TOC instruments measure TOC. So TOC sensors are normally referring to that direct conductometric method, while TOC analyzers typically refer to membrane conductometric. So with that in mind, TOC sensors, as we had chatted about, experience over and underreporting from ionic interference, and also don't have the ability to differentiate between inorganic carbon and total organic carbon, other than the use of an algorithm. So it means that TOC sensors, struggle with the USP 643 compliance. And then from an RTT perspective, the guidance document that we had referred to, ASTME 2656, requires that online analyzers should perform equivalent or better to that than the laboratory instrument. So what that means is that the online analyzer must read lower than a measurement from the laboratory instrument at the same time point in which that online instrument is reading to where you grab or isolate a sample. So that's typically a fairly easy standard to pass, given that there's total organic carbon intrinsic to the atmosphere, which can contaminate those laboratory samples as you isolate them. But given the potential for over and under reporting with sensors, it may be more difficult to meet this criteria. So with those factors in mind, it's not recommended to pursue RTT with existing TOC sensors. While they're great for monitoring and control purposes to maintain a pulse on the water quality, when it comes to leveraging an online instrument as the instrument of record for release, analyzers will provide the necessary confidence in doing so.
Great. Thanks for that informative response. That was very detailed. Briana, we'll go to you. Are rapid bioburden methods validated and accepted by regulatory agencies?
Great question. So yes, some rapid bioburden methods have been validated for specific applications by end users. And they're generally deployed as a monitoring method in conjunction with compendial testing. So the reason why we still do compendial testing is this allows for the accumulation of data and results and the ability to compare correlation, and then at the very end have a decent amount of data to have new acceptance criteria.
Awesome. Thank you. We are down to our last question. So if you have any that you would like to ask, please go ahead and submit them now, as we do still have a little bit of time left, and we'll make sure to get those covered. Hayden, this last question is for you. How much sample volume and how much LAL reagent volume is needed for each sample replicate with the Sievers Eclipse?
That's a great question. So every sample you pipette onto the Eclipse microplate, you only need to pipette one time, and that amount of sample you need is 55 microliters. So that 55 microliters is going to split into 4 replicates. 2 replicates will be your sample, and the other 2 replicates will be your sample with a positive product control. The amount of LAL reagent you need for the full microplate is just 1 milliliter, and you pipette that one time at the end, and what that's going to do is it's going to distribute equally to all your samples in an optical well and take your reading over time for that color change.
Wrap up the presentation for today. If you do have a question after the presentation, the speaker's contact information is on the screen. You can also submit it in the on-demand portion of the webinar. Thank you, Kaitlyn, Hayden, and Briana for your time today and your presentation. We would also like to thank LabRoots for hosting. Before we go, I'd also like to thank our audience for joining us today and for the interesting questions. Any questions that we did not have time to address today, or those submitted during the on-demand period, they will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.